People in Nevada were shocked when millions of bees were released into the desert. At first, many laughed, not knowing what was coming. Some even said the plan would fail within weeks. But as time passed, the story took a surprising turn, leaving people silent and experts racing to explain what happened. But the real twist wasn't just what happened in Nevada. It began decades earlier, with a bizarre plan involving tiny flies and fire ants. Why would scientists even try this? How the idea started. Red imported fire ants carry the scientific name Solenopsis invicta. They arrived in the United States in the 20th century and spread through the southeastern states. Farmers, utility workers, and homeowners all saw the same problem. Young poultry and small livestock sometimes died after severe ant attacks. Sometimes, ants were found inside electrical equipment, which caused malfunctions and outages. Parks, sports fields, and schoolyards are filled with round ant mounds. Chemical baits and sprays helped where they were applied, but they were expensive and hard to use across whole counties and regions. That problem pushed researchers to look for a different option. During the 1980s and 1990s, a team of entomologists planned a new approach. Scientists from the United States Department of Agriculture Agricultural Research Service, the University of Texas at Austin Brackenridge Field Laboratory, and Texas A&M University joined with entomologists in Argentina and Brazil. They traveled to the ants' native range to search for natural enemies. Field collectors found several tiny flies that attack fire ant workers. The flies belong to the genus Pseudaction. Specialists showed how the flies moved and hunted along ant trails. Field observations in Argentina and Brazil gave a clear picture of how the flies acted. Adult flies hovered near foraging trails and often struck a worker. The attacked worker left the line of ants and moved into the shade or into a crack in the soil. The fly laid an egg or deposited a larva that developed inside the ant. Over days, that larva killed the host worker. Researchers also saw a second effect in the field. Where fly populations were common, fewer workers left the nest to forage during the day. That change suggested the flies did more than kill a few workers. They also altered ant behavior in a way that could reduce food gathering. Turning the natural history into a control method required careful testing. United States laboratories brought candidate Pseudaction species into quarantine and exposed them to a range of North American ant species. The purpose of those trials was to show that the flies preferred Solenopsis invicta and would not attack native ants. They recorded which ant species the flies approached and whether they attempted to lay eggs. Only fly species that showed a strong preference for the invasive fire ant moved forward in the program. The testing aimed to avoid harm to native insects. Regulatory review followed the tests. State agricultural departments and federal agencies required formal release plans. Researchers prepared detailed protocols that described how they would rear flies, how they would transport parasitized ants or adults, and how they would release them on field sites. Permits set limits on transport and required steps to prevent accidental escape during handling. Extension agents and university outreach staff briefed landowners and officials so everyone at release sites knew what to expect. Technicians then built rearing programs in secure facilities. Laboratories kept fire ant colonies healthy and fed so flies could continue their life cycle. Staff learnt to handle parasitized workers without harming the developing fly. Release methods included placing small cages of parasitized ants on active mounds and releasing lab-reared adults along known foraging paths. Field crews logged every release with the date, the species name, the number released, and the exact location. They recorded weather and habitat features because temperature and humidity affect fly activity. Field releases began in the late 1990s. The first reported establishment of Pseudaction tricuspis occurred in parts of Florida. Researchers such as S.D. Porter and L.W. Morrison wrote reports on initial establishment and on how flies spread from release points. Programs in Texas added Pseudaction cavatus and other species to match different fire ant populations. The effort did not aim to remove fire ants in one season. The goal was to add a natural enemy that would change ant behavior and reduce foraging pressure over the years. These early steps set the pattern for decades of careful releases and monitoring across multiple states. International partners continued to offer guidance, and funding supported fieldwork and laboratory studies. But while the field trials hinted at success, the real mystery unfolded in locked labs. How did scientists actually smuggle a parasite of fire ants into America without disaster? How the flies were brought here. When the first candidate fly species passed quarantine tests, the work moved into secure rearing rooms. Researchers from the United States Department of Agriculture Agricultural Research Service, the University of Texas at Austin Brackenridge Field Laboratory, and Texas A&M University set up programs to raise, transport, and release pseudaction flies. Partners in Argentina and Brazil continued to provide field guidance and source material for the fly species that matched local fire ant types. Rearing phorid flies is delicate work. Labs kept live fire ants to feed fly larvae and to supply workers for parasitism. Technicians learnt to spot parasitized ants by the small bulge that forms where the fly pupa develops. Some facilities used an automated attack box and humidity controls to keep conditions stable while flies completed their life cycle. Staff fed ant colonies sugar and protein and moved parasitized workers carefully to reduce accidental deaths. They used two main release methods. One method placed small cages of parasitized ants directly on active fire ant mounds. The cages let adult flies emerge near others so the flies could find hosts on their own. A second method released adults reared in the lab near foraging trails or mound edges. The change in ant behavior altered local competition. In plots with persistent fly activity, native ant species sometimes increased their presence at baits and along trails. With less fire ant activity, other species could take small food items and occupy microhabitats once held by fire ants. Researchers recorded shifts in seed removal and in predation on small arthropods. Those effects depended on habitat and the mix of native species at each site. Some places showed an increase in native ants foraging where phorid flies had been active for seasons. Long-term studies tracked colonies for months or years. In some cases, researchers found slower colony growth, lower brood production, or smaller worker size where flies were abundant over multiple seasons. In other cases, colonies showed little long-term decline because they adjusted their behavior or accessed food during low fly activity. Disease, weather, and other predators also affected colony change, so teams kept careful records to separate the effect of phorid flies from other causes. Experimental designs used paired plots and repeated measures to show cause and effect. Scientists compared similar plots with and without fly releases and measured bait removal, worker numbers at stations, and brood development. They used traps to catch adult flies and counted parasitized workers. Those paired studies produced consistent results about behavior, repeated fly attacks, lower foraging, and force behavior change. Whether those changes led to colony failure depended on many local factors recorded in the trials. Field reports by researchers such as S.D. Porter and L.W. Morrison provided concrete numbers in trials. In monitored plots, bait removal fell by measurable amounts during active fly periods, and native ants increased activity in plots. Other researchers reported seasonal peaks in parasitism and clear links between repeated fly pressure and slower brood growth. These empirical records helped build the long-term datasets that clearly show how behavior change develops over time. But numbers on a page only told part of the story. The real test came with time, because if flies could reshape behavior in weeks, what might decades reveal? What changed over decades? After the first releases, the program expanded along the Gulf Coast and into the Deep South. Scientists from the United States Department of Agriculture Agricultural Research Service, the University of Texas at Austin Brackenridge Field Laboratory, and Texas A&M University coordinated many releases. They worked with state agricultural offices and with university extension services. Scientists added more species of pseudaction to match different fire ant types. Early efforts concentrated in North Florida and East Texas. Today, established fly records include many counties in Florida, Texas, Louisiana, Alabama, Mississippi, Georgia, and South Carolina, where monitoring shows recurring captures and sustained presence in warm seasons. Researchers keep mapping the spread and planning new targeted releases. Field monitoring over the years showed clear patterns. In some counties, Pseudaction tricuspis and Pseudaction curvatus established breeding populations that spread outward from release points. Natural dispersal moved flies at rates that changed with weather and with the type of land cover. Flies spread faster in warm, humid areas where adults survive and reproduce well. Cold winters and dry spells limited numbers in northern and inland pockets. Those limits produced a patchwork of strong and weak zones across the southern landscape. Matching fly species to the local ant population proved important. Fire ant colonies differ by social form and by worker size. Polygyne colonies have many queens and many small workers. Monogyne colonies have one queen and larger workers. When researchers released a Pseudaction species that preferred a particular worker size, the chance of establishment rose. Researchers used surveys to determine the dominant colony type and chose fly species accordingly. South American collaborators continued to supply candidate flies and field knowledge. Effects on other insects and on small food webs were noticeable in monitored plots. In areas with persistent fly activity, bait removal by fire ants fell, and native ant species often increased their activity at baits. Seed removal by native seed eaters rose in some plots where fire ant foraging declined. Predators such as ground beetles and small birds showed small differences in where they foraged in sites with many fly attacks. Those effects varied with habitat and with the local mix of insect species at each site. Long-term trials asked whether flies could lower fire ant numbers at the landscape scale. Results varied by place. A single Pseudaction species released at one location rarely removed fire ants across a whole county. Where scientists released several fly species over time and where other natural enemies or local control measures were present, researchers saw larger and more persistent reductions in foraging pressure. In some sites, the combined pressure from multiple flies and occasional baiting produced clearer reductions in colony growth and brood production over several seasons. Methods improve with experience. Scientists standardized trap designs, sampling intervals, and data recording so that results from one state could be compared with those from another. Rearing techniques became more reliable, producing cohorts of adult flies timed for release during peak ant activity. Researchers began to use global positioning system coordinates for release mounds and to log weather conditions at the moment of release. These steps made it easier to track the spread and to evaluate which release strategies gave stronger establishment. Funding and partnerships sustained the work. Federal research funding supported core laboratories. State agencies and university extension programs provided field staff and local outreach. Private landowners and pest control firms sometimes hosted release sites or shared local observations. International collaboration with entomologists in Argentina and Brazil remained important for locating new Pseudaction species and for sharing field knowledge. The program depended on steady cooperation among many institutions and individuals. Let us know your thoughts in the comment section below. And don't forget to like, share, and subscribe for more interesting videos.